Hello everyone, I'm Harshada.
And I am Radhika. We are the students of Rajaram College, Kolhapur.
We are going to tell you about biofertilizer and its application.
Biofertilizers are the organisms that enrich the nutrient quality of soil. The main sources of biofertilizers are bacteria, fungi, and cyanobacteria, that is blue-green algae. Biofertilizer refers to the use of soil microorganisms to increase the availability and uptake of mineral nutrients for plants. Biofertilizers can be defined as a live microbial preparation when applied to seed, root, or soil, colonizes the rhizosphere or the interior of the plant, and promotes growth by increasing the availability of primary nutrients and by releasing growth promoting substances. Nowadays, instead of biofertilizers, the term microbial inoculants is commonly used. Rhizobium is a bacteria. It is present in soil as well as in root nodules. It carries out nitrogen fixation, that is, nitrogen gas is converted into ammonia. Ammonia is utilized by leguminous plants. Rhizobium, there is swollen part of root which is called nodules. The plant bearing nodules are called leguminous plants. The nodules consist of thousands of rhizobium cells. Nitrogen enters in nodules from environment. Nodule can be hence said to be factory of ammonia. The association between rhizobium and leguminous plant is called a symbiotic association. Sources: any soil, especially soil with leguminous plants and root nodules.
Of rhizobium biofertilizer requirements are enzyme nitrogenase, energy. Nitrogen fixation is highly energetic process, hence 12 ATP molecules are required. It is intracellular process. Energy source is sugar molecule, that is sucrose. Development of anaerobic condition is required because enzyme nitrogenase is oxygen sensitive. Production of rhizobium involves various steps. From that, first is selection of strain. Here, rhizobium strain is to be selected for commercial production. It can be done by either purchasing or by isolation. The strain can be purchased from culture collection center, or we can isolate the strain. For isolation, we will have to take soil or root nodules as a source. For that, first collect the leguminous plant. Wash the root nodules by taking it under tap water. Collect the nodules from roots on the petri dish. Then add water into petri dish so as to wash root nodules. With forceps, take nodules in another petri dish. Take single nodule and add it to sterile saline aseptically. Open test tube aseptically and crush the nodules in sterile saline with sterile glass rod. The crushed nodules, then we will make suspension of it. Streaking of nodule, suspension of sterile Congo red yeast extract mannitol agar plate by 4-quadrant method loopfully. Label the plate. Incubate it at room temperature for 24 to 72 hours. Now observe for colonies. If no colony appear, then we will have to incubate it further. Pale color colonies are to be selected and red color colonies are to be rejected because Congo red indicates contamination in Crima medium. It is a differential medium. Rhizobium don't absorb Congo red, hence they are pale color. After isolation, gram staining and motility is performed. Maintenance of the strain to preserve the best organism obtained in live form is essential. This can be done by refrigeration or by lyophilization. Next is mass production. This is performed by two steps: inoculum preparation and second is mass production. In inoculum preparation, take a slant 250 ml of yeast extract mannitol broth medium for cultivation is used. Inoculate the microorganisms. Incubate them for 24 hours at room temperature. Again increase the medium capacity to 2 liters and add those organisms to medium and repeat all the procedure. In mass production, prepare a medium in a flask or fermenter. Then take organisms from prepared inoculum and add to huge flask of about 5 liters or directly in fermenter. Shaker is needed for incubation of organisms. Shaker provides aeration. Incubate the organisms for 24 hours at room temperature. This is nothing but liquid biofertilizer. Separate the broth and organism. Biofertilizers are available in two forms: liquid biofertilizers and solid biofertilizers. Liquid biofertilizer is to grow microbes in suitable medium up to particular limit. Take sterilized bottle for packing with different quantity. Pack bottles tightly and label them. Preserve the bottles at low temperature. Solid biofertilizer. It is carrier-based inoculant. Here, selection of carrier is done, then the removal of unwanted materials is done by hand or by sieving procedure. Pulverization and neutralization of carrier is done, and then sterilization. Broth produced at mass production is taken and mixed with sterile and processed carrier. Spread this mixture on paper and this is kept for 2 to 5 days. This process is called curing. Keep this mixture away from sunlight or in shade or in room at about 20 degrees Celsius. Then purpose of curing is to adjust organism with carrier material, because organism grow during 2 to 5 days, that is during curing. We will have to first take 1 gram of mixture, then perform SPC from day 1 to day 5. If the count is increasing, then the carrier is suitable for organism. Count of organism is expected to be increased. Now the biofertilizer is ready. Carrier. Carrier is a solid inert material. It acts as a support to leave or to solidify. Some of the carriers are peat, lignite, charcoal, IPS, that is Indian peat soil, etc. Criteria for selection of carrier material is: it must be cheaply and easily available, it must be non-toxic in nature or should not harm any of the organism, it should have good absorption ability, it must have more shelf life, it must have good moisture holding capacity, it must be easily pulverizable and sterilizable, it must have good addition with seed, it should have high organic content. Packaging, labeling and storage. Packaging: in different quantities it is packed in polythene bags like 250 grams, 500 grams, 1 kg, 5 kg, etc. Labeling: points to be labeled: first, name of the manufacturer, then second, date of manufacturing and expiry, third, storage conditions, type of crops to be used, type of carrier present, amount of product or net quantity, method of application, price of the product, quantity to be added. Storage. Storage is done at cool temperature. Method of application. First, seed application. Prepare slurry of biofertilizer. Slurry means biofertilizer plus water. Mix the seeds with slurry. Seeds are coated with biofertilizers. Seeds are removed from the container. Take it and place it on paper for about half an hour or 1 hour. Procedure is to be carried out in the shade. Let the seed dry. Seeds are ready to sow. Coated seeds are inoculated. The method is conventional. For binding of biofertilizer with seeds, some sticky substances like jaggery gum is added with the slurry. Seedling application. First, prepare solution of slurry plus jaggery. Dip the seedling's root in the solution above for about half an hour. Remove the seedlings from solution and plant it. Along with rhizobium, we can use azotobacter and azospirillum as nitrogen fixing biofertilizer, but those can be used for non-leguminous plants. Along with nitrogen fixation, phosphate solubilization is also essential, hence PSB and VAM can be used. PSB means phosphate solubilizing bacteria and VAM, that is vascular arbuscular mycorrhizae, can be used. So we have told you about biofertilizer. Thank you.